Great, good afternoon, welcome back. As mentioned, I'm Erica Barnell. I'm from Washington University in St. Louis, an MD PhD candidate there where I'm completing my PhD in Obie Griffith's lab. And today I'll be talking about somatic variant refinement and the ability to use a deep learning approach as well as a standard operating procedure for that process. Disclosure statement, I have a startup company. So the annotation bottleneck has been covered extensively throughout this conference, specifically the process by going through sample procurement, nucleic acid extraction, library preparation, and parallel sequencing is very automated with high throughput. But once you go to alignment, somatic variant identification, further refinement of somatic variants to eliminate false positives, and then clinical annotation, it is extensively laborious, and there are too many variants to annotate in a meaningful way where clinicians can understand that data and act upon it effectively. Today I'll be describing two tools that we developed in the Griffith lab. The first is a machine learning approach to eliminate false positives associated with existing somatic variant callers. And the second, we recognize while manual review is incredibly laborious, it still has a role in this annotation process, and therefore we've defined procedures and methods that we believe are optimal for identifying which variants are true positives and which variants are false positives. The first tool is DeepSVR, or Deep Learning Somatic Variant Refinement. The entire data set and cohort is available online, and I'll be going over the brief development methods and initial validation of this deep learning model. For model development, we obtained a training data set. This consists of 59 different features, most of which are sequencing features that are derived from tumor and normal BAM files. It includes the variant allele frequency, tumor depth, mapping quality, things like that. We built a deep learning model using the training data, and then we employed that model on a cohort of testing sets. All of those had, again, 59 features that were associated with the sequencing data. The predictions from the model was an array of 3 predictions, including somatic, ambiguous, or failed calls. And then finally, we developed the prediction accuracy by comparing the model performance to orthogonal sequencing or to original manual review calls. This is the cohort that we use for training and testing of the model. Specifically, we had 41,000 variants that were obtained from 9 different tumor subtypes. This represented over 400 cases, each of which had tumor and normal sequencing, and all of which had manual review calls for understanding model performance. The variant calls were parsed by somatic, ambiguous, failed, or germline, and ultimately the germline and failed calls were collapsed into a single negative failed finding, since this is a somatic variant caller. We looked at 10-fold internal cross-validation to assess initial performance of the deep learning model, and that performance parsed by call type is shown here. For the failed and somatic calls, we attained a ROC AUC of greater than 0.96, which is excellent. The ambiguous calls, for variant calls that were identified by reviewers as ambiguous, there was a slight reduction in the accuracy, which is likely attributable to the inter-reviewer disagreement for those types of calls. Subsequently, we looked at 2 independent testing sets to evaluate model performance. The first is shown here. So we derived sequencing data from a very unique cohort. Specifically, it was an AML31 case where we evaluated or identified variants using 7 different variant callers. In total, there were over 200,000 variants identified by any one given caller. And then we subsequently did ultra-deep sequencing on each one of those genomic loci to evaluate if it was a true somatic variant or a false variant that was attributable to a sequencing artifact. For all 200,000 variants, we employed the deep learning model on this cohort, and you can see the resulting AUC was 0.95, demonstrating that we were very capable at recapitulating the orthogonal sequencing results for this cohort. The second independent testing set we used was from the TCGA. So we obtained 108 cancer patients from 8 different tumor subtypes. These are shown here on the x-axis, and we parsed out the number of true positives and false positives that were demonstrated by orthogonal sequencing. The resulting ROC AUC for each one of these cases is shown on the y-axis, and that you can see for most of these cases, we very accurately recapitulated the orthogonal sequencing results. However, there were some cases in specific tumor subtypes where there was a reduction in the total accuracy. When you concatenate all these results, the ROC AUC was significantly lower than our original validation cohorts of 0.78, and we attribute this reduction in AUC to the methods by which the TCGA called false positives. So they were using methods that were not used in our training cohort for calling false positives, so we hypothesized that retraining the model with incremental amounts of the TCGA data would restore the accuracy of the model. This experiment is shown here, where the x-axis represents the total number of variants that were used to retrain the deep learning model, and when you add up to 400 variants from the TCGA cohort, we recapitulate the model accuracy and restore model performance to an AUC of 0.9. Methods for downloading this software, for building the model, and for incorporating your own data into the model for development have been provided online in the GitHub wiki, and if you have any questions on how to go through that process, please feel free to ask. Finally, I mentioned that the deep learning model can eliminate up to 90% of all false positives from automated somatic variant callers, but we still recognize that many of those variants are identified as ambiguous using the deep learning model, and therefore we recommend using the manual review SOP, or standard operating procedure, for evaluating those variants. This SOP first defines an integrative genomic viewer for evaluating these variants. Specifically, we take IGV and we break it down into 3 distinct parts: the genomic ruler, the data tracks where you actually visualize the data, and the genomic features. We then further define some features within IGV that help with the manual review process. We also built an applet plugin called IGV Navigator, which is displayed here. This is the IGVNav interface, and B shows the IGVNav input file. Specifically, this is a bed or bed-like file that contains 8 columns. The first 5 columns are the genomic loci being evaluated, and the second 3 columns are blank and will be annotated using IGV. When you go through the manual review process, all of the genomic loci are displayed in the IGVNav interface. The user then can subsequently go through the manual review SOP, label the variant as somatic, germline, ambiguous, or fail, and then specifically for calls that are labeled as ambiguous or fail, we recommend that users annotate those with one of these 19 sequencing artifacts that are commonly observed during IGV review. These include things like low mapping quality, high discrepancy regions, if there's an adjacent indel, things like that. This is how this can be annotated. For features that are not predefined in IGV, the user can also add optional notes in a free text form, and those are, again, displayed in the output file here, where you can filter on the call, the tag, or the notes. So as mentioned, we define this process for setting up IGV, for setting up IGVNav, and for loading the feature tracks, and that takes 6 discrete steps, which a cheat sheet is described here. And then subsequently, for each variant that requires manual review, we set up a 7-step process to evaluate for each one of those 19 artifacts that are commonly observed during the process. Finally, we created this extensive supplement where we define each of the 23 selections on the IGVNav interface. Each selection has an example and a description of the specific feature, as well as helpful hints for identifying those specific types of variants or tags. Finally, we validated this SOP by evaluating variant calls and asking novice manual reviewers to review variants before and after reading the SOP. So specifically, we had 4 novice reviewers. 200 variants were reviewed before reading the SOP, which is shown in light blue, and then 200 variants were reviewed after reading the SOP. To assess improvement in performance, we evaluated reviewer accuracy of these calls when compared to orthogonal sequencing data. We evaluated the inter-reviewer agreement between calls, and we also evaluated the amount of time that it took for individual review to be performed. The top two panels show the improvement in the ability for reviewers to identify somatic variant calls. So as mentioned, the light blue represents the review percentage before reading the SOP, and the dark blue is after. So for each of these 4 reviewers, there was an increase in the total percentage of somatic calls being identified, and that was significant when you concatenated all 4 individuals together. Subsequently, there was an increase in the inter-reviewer agreement between all reviewers, and although there was not a significant reduction in time, what I thought was interesting that there was a reduction in the variance of the amount of time, and I think that again is attributable to providing a discrete steps for individuals to go through for each variant. The limitations and future direction for both of these tools include we want to add use cases for data without manual review calls. So currently you require to have manual review calls to understand the accuracy of your data, but we'd like to incorporate other methods of validation. We want to build multiple models for additional variant types. So currently we only evaluate SNVs and indels, but obviously there are other different types of variants, so it would be important to incorporate those. And then finally, it's been requested that we have a tumor-only analysis. So we hope to build a subsequent model for tumor-only, and then also look at a manual review SOP for tumor-only samples. And then I would be remiss if I didn't say that Elaine Martis really inspired this work. In 2010, I believe, she implored us to understand how we avoid the pitfall of having a cheap human genome, but complex and expensive manual analysis to make clinical sense of the data, and I really hope that these tools address that issue and help us better understand how to overcome these sorts of pitfalls associated with precision oncology. With that, I would love to thank my lab. I have no background training in computer science, and so most of this could not have been possible without their help. Thank you.